Hello everyone and welcome back to my channel Micro and Mol Bio with Dr. Sunita Bundale. In the previous lectures, we have talked about nucleotide excision repair and base excision repair. In today's lecture, we will be talking about yet another excision repair pathway, and that is the mismatch repair pathway, a pathway completely dedicated to repairing the mismatches that happen at the time of replication. The action of DNA polymerase III, including its proofreading activity, results in one misincorporation per 10 to the power 8 bases synthesized. Some mismatches due to replication errors escape proofreading. Mismatch repair reduces this rate to one chain in every 10 to the power 10 or 10 to the power 11 bases, thus increasing the accuracy of DNA synthesis by 2-3 times. MMR recognizes mismatched bases in DNA, for example, G against a T or A against a C base pair. These do not cause large distortions in the helix. The mismatch repair system apparently reads the sequences of the bases in the DNA. Most mismatches are due to errors during replication. However, mismatches can also be produced by other mechanisms, for example, by deamination of 5-methylcytosine to produce thymidine, improperly base paired to G. Regardless of the mechanism by which they are produced, mismatches can always be repaired by the mismatch repair system. This repair system has to work quickly, otherwise the mutation would get fixed and the repair system would not have any idea about which was the mispair. For example, this is a double-stranded DNA molecule which is undergoing its first round of replication, and during replication in one of the duplexes you can see here that against an A, a G was incorporated. So this is the mispair here. In the second round of replication, against this G, a C would be added. And now the repair system would have no idea which of them is the wrong base pair that has been incorporated. We say that after the second round of replication, the mutation has been generated or the mutation has been fixed. Hence, the mismatch repair system has to work before the second round of replication, which means that before the mutation has been fixed. The mechanism of mismatch repair system was first thoroughly studied in E. coli. The proteins that initiate the repair process are MutS, MutL, and MutH. MutS is responsible for initiation of E. coli mismatch repair. It is a 95 kilodalton polypeptide which exists as an equilibrium mixture of dimers and tetramers and recognizes mismatched base pairs. MutL, a 68 kilodalton polypeptide that is dimeric in solution, is recruited to the heteroduplex in a MutS and ATP dependent fashion. MutS, MutL heterocomplex is believed to be a key intermediate in the initiation of the mismatch repair. MutH is the endonuclease that brings about the nick. In the figure, the replication error produced mismatch is indicated by the distorted double helix. You can see that the distortion here is because of the mismatch that has occurred during replication. MutS scans the newly replicated DNA and recognizes such mismatches due to the distortion they cause in the DNA backbone. The mismatches are true mismatches plus loops caused by insertions or deletions of up to 4 nucleotides. And it binds to the mismatch containing DNA, inducing a pronounced kick in the DNA and a conformational change in the MutS itself. The next step is where MutL now binds to the MutS DNA and stabilizes the complex. The MutS MutL complex activates the next enzyme, MutH, to come to the site, which binds to the parental strand. It is a U-shaped enzyme which sits on the parental strand and crosses its arms so as to cause a nick in the daughter strand. The nick can be introduced either on the 5' side or the 3' side of the mismatch. Depending on the nick site, whether it is 3' or 5', different exonuclease is recruited and it is responsible for removing a few nucleotides along with the mismatch. As a result, a gap is created which is filled by DNA polymerase, which binds to the 3' OH end and uses the parental strand as template to synthesize the DNA. Once the gap is filled, the nick is sealed by ligase and the mismatch is repaired. The major challenge faced by MMR is to correctly recognize the newly synthesized daughter strand that has the mistake and leave the parental strand alone. How does E. coli mismatch repair system know which of the two mismatched nucleotide is to be replaced? At least in E. coli, this is not a problem. This is because E. coli genome contains palindromic GATC sequences every 250 base pairs. The adenines in these GATC sites are methylated. GATC is a palindrome, so the opposite strand also reads GATC in the 5' 3' direction. This means that a newly synthesized strand across from a methylated GATC is also destined to become methylated. This methylation is brought about by DAM methylase, which closely follows the newly replicated strand. It takes approximately 1 minute to reach the methylation sites. For this 1 minute, the parental strand is methylated and the daughter strand is non-methylated. E. coli MMR exploits this difference in the two strands to correctly recognize the daughter strand from the parental strand and correct the mismatch in the daughter strand. This process must occur fairly soon after the mismatch is created, or both strands will become methylated and no distinction then will remain between them. E. coli DNA is normally methylated at GATC sites, just like I told you now, but the newly synthesized strand is not immediately methylated. The fact that the old strand but not the new is methylated near the replication fork allows E. coli cells to distinguish the old strand from the newly synthesized daughter strand, which has the mispair. We have seen that different exonucleases can be used to remove the single-stranded DNA between the nick, which has been created, the MutH, and the mismatch. MutS recognizes the mispair and MutL stabilizes it. MutH binds to the nearest methylated adenine on the parental strand. We have seen this just now. If the nearest methylated adenine is on the 5' side of the mispair, MutH brings about a nick on the 5' side of the mispair. In this case, a 5' 3' exonuclease is recruited to remove the oligonucleotide along with the mispair. DNA polymerase binds to the 3' OH end, fills the gap, and the ligase seals the nick. On the other hand, if the nearest methylated adenine is on the 3' side of the mispair, MutH brings about a nick on the 3' side of the mispair. In this case, a 3' 5' exonuclease is recruited to remove the oligonucleotide along with the mispair. DNA polymerase then binds to the 3' OH end, fills the gap, and the ligase seals the nick. It is important to note that the use of methylation to distinguish the parental strand is probably peculiar to E. coli. Data from yeast and vertebrate in vitro mismatch repair experiments suggest that single-strand nicks provide a signal for strand specificity in these organisms. Note that single-strand breaks are present in nascent DNA strands between Okazaki fragments in the lagging strand and at the 3' end of the leading strand. Thus, this method of recognizing the daughter strand from the parental strand by methylation is present in prokaryotes, but it is absent in eukaryotes. Eukaryotes recognize the daughter strand from the parental strand because of the fact that the daughter strand, the lagging strand, is formed in the form of Okazaki fragments, and there are several nicks present in the daughter strand. Human homologues to MutL and MutS have been discovered because mutations in them can cause one of the most common hereditary cancers, the hereditary non-polyposis colon cancer. Whereas MutS and MutL function as homodimers, the eukaryotic proteins function as heterodimers. Heterodimers of MutS homologues are responsible for initial recognition of the mismatches and small insertions or deletions, and heterodimers of MutL homologues interact with the resulting complex, as in the case of E. coli. In contrast to E. coli, however, some eukaryotic MutL homologues play a specific catalytic role in mismatch repair. The eukaryotic MutL homologues have an important endonuclease activity. Two heterodimers of MutS homologues are found in human cells. One of these dimers is called MutS alpha and the second is called MutS beta. The first heterodimer preferentially recognizes single base mismatches and small incision deletion loops of up to 1 to 2 bases. The second heterodimer primarily recognizes larger loops of 2 to 10 bases. Most, that is almost 80 to 90% of the MSH2 in the cell is complexed with MSH6 to form MutS alpha. Thus, MutS beta is a relatively minor activity. Structural studies of MutS homologues suggest that MSH2 resembles the B monomer of the MutS homodimer, while MSH3 and MSH6 resemble the A monomer. Three MutL homolog heterodimers are known. MLH1 is one of the dimer subunits in every case. The major heterodimer, MutL alpha, consumes about 90% of MLH1 and contains PMS1 in addition to MLH1. The second heterodimer, MutL beta, consists of MLH1 and MLH2 in yeast as well as in humans. Note that human PMS2 is a better homologue of yeast PMS1 than is human PMS1. The third dimer, MutL gamma, consists of MLH1 and MLH3. The first dimer plays a major role in mismatch repair. The role of MutL beta is not clear. MutL gamma plays a minor role in mismatch repair system. Thus, in today's lecture, we have talked about mismatch repair system in prokaryotes, taking E. coli as the model organism. And we have seen how the E. coli mismatch repair system recognizes the mismatches and repairs it using the method of methylation to recognize the daughter strand from the parental strand. The eukaryotic mismatch repair system is slightly complex with heterodimers present in the instead of monodimers, and this particular mismatch repair system is responsible for removing the mismatches that have been created during the replication process. With this, we come to an end of this lecture. If you like the content of this channel, please like, share and subscribe to the channel. In the next lecture, we will be talking about the SOS response and the double-stranded break repair in eukaryotes. Till then, thank you and have a great week ahead.